welcome back to Science of Medicine. As we begin our physiology overview, we'll talk some about the integumentary system or the skin, a little bit on muscles and bones, although in our next block we'll talk more detailed about these. This is just to give you some basic ideas about physiology. So we can approach this through a systems-wide approach where we deal with different things such as the kidneys and or the blood, and that's the approach that we'll actually use across this block. So if we look at the skin is a very complicated structure, and it occupies a large portion of our body, and it's comprised of three different layers. The first is the epidermis, which lies on top of the dermis, the second layer, and then underneath is that subcutaneous layer. There are many layers in the epidermis. The uppermost layer is dead, and the lower layer is alive. The skin cells will grow and be pushed upward and will last for about a month. Now, it's very important to note that between the epidermis and the dermis, there is kind of a corrugated layer that basically has the dermis pushing up into the epidermis. And this is used so that when you have a shear force that is applied across the epidermis, it doesn't just slough off. So it provides for mechanical stability. Now within that epidermis and dermal layer, this is where we'll many times form blisters, for example, that will well up. Now that bottommost layer of epidermis is alive, as I mentioned, and one of the things that's important is that those cells will actually extend down into the hair follicles. And in fact, this is very important because if you have a burn that is very severe, one that actually kills a lot of the epidermis, as long as those hair follicles remain alive, there are epidermal cells down there that can regenerate and regrow your skin. Nowadays, we could transplant skin from one area on the body to another, but we also can grow this externally. Now, in the dermal layer, there is a lot of structures. First off, different types of cells. There's the erector pili, smooth muscle, which are involved in having our hair stand on end when we're cold, so that it creates a stagnant layer of air to hold in that heat. There are also nerve and blood vessels that are in there, as well as glandular tissues that will secrete the sweat that we have. We have many different types of immune cells that are in there, particularly the mast cells and macrophages. And in the lower layer, in the subcutaneous layer, we have additional cells that are down there. In general, most of us have experience with the epithelial layer, although sometimes we'll actually slough that off or braid that and get down to the dermal layer, which is actually quite painful if we do that. If we were to burn off the epidermis and the dermis and actually go down even further, we actually would not feel pain because we would burn off those nerve endings and the sensory cells for being able to do this. And that's very important for people that have full thickness burns. Now, what does our skin do? It does several things. One of the primary functions of this is to protect us. We are made mostly of water, and as a result of this, we would desiccate, or that water would evaporate. And so we need some sort of protection, and that's in the way of this hydrophobic outer skin layer, which is dead. And so as a result of that, the permeability to substances is limited to those that are very lipophilic, such as your fat-soluble vitamins A, D, E, and K, as well as steroid hormones. And in fact, this is how you will get a lot of steroid hormones such as androgel or testosterone, or if there is a contraceptive patch, you can have things such as estrogen that will actually be transmitted through your skin. It also is there for protection for the various milieu of fungi as well as bacteria that are inhabiting the surface of our skin. Our skin protects us in other ways. We have the ability to sense our environment through our skin. Pain, pressure, and temperature are the typical ones that we talk about. And we have different densities of those sensors. For example, our hands have a much higher density than those on our back. We do make vitamin D through the action of our skin and sunlight. And we also have thermal regulation primarily through evaporation, but other mechanisms that we'll talk about in a second. So in order to provide for the biochemical processes that need to go on inside my body, I need to have a fairly well-regulated thermostat. And I have sensors within the core of my body, in particular things like my spinal cord or my abdomen or thorax. And they maintain a core body temperature between about 97 and 100 degrees Fahrenheit, which is a fairly narrow range to optimize enzymatic reactions. If that temperature is too hot or too cold, I will be able to regulate that through my skin. If I'm cold, then I can shiver, that is to move muscles back and forth without actually causing much in the way of movement, but it does dissipate or generate heat that can be used to warm me. I can reduce sweating and even vasoconstrict blood vessels so that I don't take that precious heat and move it to the external side. This is the basis for why we get frostbite. It is much more important for us to protect our core than it is to protect our fingers or toes or tips of our noses. And so with that vasoconstriction, we cut off blood supply to those digits. However, that causes necrosis if it's cut off for too long of a time. Similarly, we can dissipate heat by sweating. And when we sweat, remember, we'll take that heat of vaporization of water, which is fairly great, and lose that heat as we have the phase transition of the water from the surface of our skin from the liquid to the gaseous state. Now, I do have several sets of receptors. I have mechanoreceptors that when I have a physical deformation, it allows ion channels to open, which generates an electrical signal. Those are for detecting pressure and vibration. I have a set of thermoreceptors and nociceptors, which are for pain. These are very thinly myelinated or unmyelinated neurons that will fire at a certain rate and will be influenced by how fast or slow they fire based upon temperature or pain. And I do have some chemoreceptors that respond directly to certain chemicals such as histamine or substance P or acids. Now, in my musculoskeletal system, I'm having a number of bones. In the adult, that's just over 200. In the child, it's more than that. Many of those bones would fuse. And those bones are essential for us to be able to have a structure, not only for protection, but for also movement. Many bones will articulate into a joint between two bones, and at the articulation between those two bones, there will be cartilage from chondrocytes that are made. And I will actually have connections between muscles and bones, and I will need to slide across that joint, across that cartilaginous joint, and that's facilitated with these bursa. Now, I also have muscles, although in the musculoskeletal system, I'm only talking about skeletal muscle. For this particular block, I'll actually talk about smooth and cardiac muscles that we'll talk about when we talk about gastrointestinal tract as well as the heart. I have connections between muscles and bones. Those are called tendons, and connections between bones, and those are ligaments. If I look at muscles, I have many, many more muscles than I have bones, well over 600 that are named. and they're oriented in various directions, because all that a muscle can do, a skeletal muscle, is only shorten. And we'll talk about how that process occurs. And so I can apply forces depending upon how those muscles are oriented. So my musculoskeletal system provides structure as well as protection. I have an axial skeleton as well that will be part of my body proper, and I'll also have an appendicular skeleton, which are my appendages, my arms and my legs. I am going to be moving, and the only way that I can have movement is that I can affect a force across a joint and allow that movement to occur. Now, in addition to providing structure, protection, and movement capabilities, I also store within the bones the cells that are involved in the hematopoietic system. So I'll make my red blood cells or erythrocytes, I'll make my platelets, and I'll also make my white blood cells. And I also, in the structure of the bone itself, store calcium and phosphorus, which are essential for many metabolic processes. If I look on a detailed level of a long bone, cancellous bone, I would see a structure very similar to this. I would have a trabeculated network that provides for maximal strength along the longitudinal axis of that bone, but also minimal weight. So I will be able to have in this space a filling of other cells. In this case, this is where I'm going to put my hematopoietic cells that will make this. As a result of having this place where I will make my blood cells, this is highly vascular, so on the outside of the bone I'll have penetrating blood vessels that will go to and from this, and they will penetrate through a coating on the outside of the bone called the periosteum. And this periosteum is also innervated, and the innervation is why when you break a bone and you actually break that periosteum or cause some damage to it, it hurts. Now, the bone is made up of, in addition to the hematopoietic cells, cells that will make this bone. Those are called osteoblasts, as well as cells that will break it down, which are called osteoclasts. The osteoblasts and osteoclasts are constantly remodeling your bone based upon the stresses and strains that are actually being placed on the bone. So bone is a living organism that is adapting to you and adapting to the forces that are exerted on it. If I look at the connection between two bones at a joint, then what I'm going to have is two bones that are not directly in contact with each other, but are separated by a layer of cartilage. And this cartilage is made out of chondrocytes. Now, these chondrocytes are there to make a synovial fluid, this little blue area right here, which is a fluid that helps lubricate this joint and reduce the frictional forces between it. And that synovial fluid is primarily is made up of hyaluronic acid, which is a proteoglycan that holds on to water to allow for this low viscous solution in between, and it's contained within a joint capsule. Now, outside of that joint capsule, I will have tendons that will run across that capsule. So I have to protect that joint capsule and the tendon from the frictional forces there. And so I do that with the sheath, which is a bursa. And that bursa actually, as that tendon is moving back and forth, is going to provide for lubrication and reduction in the friction. One of the things that I'll point out about this joint is that it turns out that the cells that make up these, the cartilage actually are, for the most part, radio-opaque. So if I take an X-ray of this joint, it'll look like I can see the bone because calcium and phosphate will show up. But the chondrocytes, these cells won't really show up. And so it'll look like there's a space between them. There's actually not a space. There's actually this cartilage, which is between them, and this synovial fluid. But if you have very, very bad osteoarthritis that has worn down this cartilage, then there'll be very little space in between the bones. and that will cause a lot of pain because you'll have periosteum that will be rubbing up against other periosteum. Now on the skeletal muscle side, I have a very complicated situation. So in the skeletal muscle, if I look at, let's say, the deltoid on this person's shoulder muscle, I would find that those muscle cells are oriented in a longitudinal fashion across that muscle. And if I look at it at a very, very small scale, what I would see is that I would have mitochondria to generate lots of ATP to run this muscle, and I will have these myofibrils, which are made up of actin and myosin. Both of these protein structures are essential for me being able to move along the muscle structure, along the skeletal muscle. So at the very base, I'll have this actin filament, which is kind of like a strand, two strands of pearls together, and these myosin heads, which will be able to bind into a cleft and pivot. And this will allow me to have movement across this. And my actin filaments are fixed, and my myosins will walk. And when they do that, they'll actually pull those filaments together and shorten the muscle. Now, by shortening the muscle, the whole muscle itself will actually shorten, and you can then affect movement. It is very important that you understand how this process occurs, because there are many, many medical reasons that lead to dysfunction in this. And so I want to go through this in great detail. In order for skeletal muscle to work, I have to have some sort of a conscious signal from my brain or a reflex that is coming from my spinal cord. And that means that I have to have a nerve that is going to go out to that skeletal muscle and then take that electrical signal going down the nerve to the motor nerve unit. That is the articulation of the nerve with the muscle. And when it does this, it will affect synaptic communication. A small amount of neurotransmitter will be released from the nerve. It will diffuse across the small gap or synapse and bind with the muscle. Now, the neuromuscular junction is using acetylcholine, which is the neurotransmitter that's made in the nerve that's going to diffuse across that synapse. And when that acetylcholine acts on a muscle fiber, binds with its receptor, it will open sodium ion channels. And the concentration of sodium outside the muscle cell is greater than inside. So thermodynamically, it's a spontaneous process to allow sodium ions to rush into the muscle cell. And when that happens, it will change the potential of the inside of the cell relative to the outside. And that will then cause what we call depolarization of that muscle. And I'll talk about that in a second. Now, this will continue down the length of the muscle fiber. If I didn't have a way to get rid of the acetylcholine would keep stimulating that muscle. And so to get rid of the acetylcholine in the junction, in the synapse, I have what's called acetylcholinesterase. It's an enzyme that actually degrades the acetylcholine and allows it to be taken back up by the neuron and then recycled to make more acetylcholine. On the muscle side, once I've changed the potential inside the muscle, the sarcoplasmic reticulum, that structure that I showed you before, will actually then open up calcium channels. So the sarcoplasmic reticulum is like a big storage vessel for calcium ion. Its concentration is much, much higher inside than outside in the cytoplasm. And so as soon as that sarcoplasmic reticulum opens up, calcium ions rush out of the sarcoplasmic reticulum into the cytosol, including where there is a lot of actin and myosin. Now, in the actin filaments, there is a troponin complex that's made up of several parts. One of the parts, troponin C, you could think about it as for calcium, that will bind to the calcium that comes out. And that will cause a change in the inhibitory part of troponin that will allow for it to move out of the way. And once that moves out of the way, there will then be the ability of myosin to bind in that actin filament. And once the binding of myosin to the actin filament occurs, it will cause a pivot in the head of the myosin, and that will cause a incrementally small change or shift or decrease in the distance and in that muscle, and that walking process leads to the shortening of the muscle if it's done by many different myosin actins along that muscle. We'll then have to start this over. We'll pump the calcium back into the sarcoplasmic reticulum so that can be released again. There are also sodium-potassium pumps that will be used to restore the polarization of the muscle unit, but we'll talk about that more later on. Now, in contrast, smooth muscle also has a actin and myosin, but the process is very different. Now, smooth muscle is not part of musculoskeletal system. It's found mostly in the GI tract and in blood vessels, as well as the ability to dilate or contract your pupils. The upper 1/3 of the esophagus, the first part of the gastrointestinal tract, is striated or skeletal muscle, but after that it's smooth muscle. And this smooth muscle is made up of sheets of smaller muscle fibrils that comprise this smooth muscle, many of them in different orientations to permit for what we'll call peristalsis that we'll talk about in the gastrointestinal tract. In this case, there's no neuromuscular junction. I do not have to have a conscious decision from my brain to be able to move food along my gastrointestinal tract. Thank goodness for that, because if I did, digestion would be a very complicated process you'd have to think about. Because there's no neuromuscular junction, I have to have some way of controlling it. And the smooth muscle is under the control of both hormones. If you remember, those are small molecules that travel from an endocrine organ through the bloodstream to the target organ, as well as the autonomic nervous system, which we'll talk about when we talk about neurology. Now, instead of troponin, I have calcium and calmodulin that are impacting the movement of my smooth muscle. And calcium calmodulin will activate myosin kinase. You remember that any kinase will phosphorylate. So in this case, myosin kinase is an enzyme that phosphorylates the myosin. And when you phosphorylate the myosin, it will then allow the binding to the actin. Now, once you bind the actin and the myosin to the actin, then you'll get a pivot in that myosin head, and it will walk one little unit along the actin filament. You also have a drop in myosin, calcium calmodulin concentration, and when that happens, another enzyme, myosin phosphatase, will be activated that will dephosphorylate myosin. So there's no troponin in smooth muscle. There is no neuromuscular junction in smooth muscle. And the myosin is not ready to bind. It has to be activated in order to bind and then pivot to walk. And it also has to be deactivated. It is still modulated by calcium, and in this case by calcium calmodulin. Now, cardiac muscle shares a lot of similarities with skeletal muscle in that I do have these longitudinally actin and myosin fibrils, myofibrils, but now I have segments of muscle fibril that are connected via these small, what are called intercalated discs. And these intercalated discs are simply ways that you can diffuse ions very quickly through them to allow for propagation of the ionic signal from depolarization. Now, I also do have in cardiac muscle troponins, and in fact, damage to cardiac muscles is one of the things that I detect for heart attacks, and I detect and look for in the blood troponin. So skeletal muscles and cardiac muscles have that myosin, actin, as well as troponin, unlike the smooth muscles, which has the calcium calmodulin. I don't want to think about having to have every single heartbeat, otherwise I would die very quickly because I would lose concentration and that would not be good. So instead, I have these pacemaker cells that are intrinsic. Now, the ones that normally, under healthy conditions, will set the rate at which my muscles are contracting in the heart are located in the sinoatrial node of the right atrium. When we talk about cardiac physiology, we'll talk more about those. But they will actually be set to fire off automatically about 1 time a second. They initiate that depolarization. There's no acetylcholine. They just simply open up their channels and this happens. I can modulate that through the sympathetic nervous system to increase the heart rate, or the parasympathetic nervous system to slow the heart rate. I also have other pacemaker cells in different parts of the heart in case those SA node pacemaker cells for some reason don't work or their signal gets blocked. Now, I do have sodium channels like I have in skeletal muscle, but I have 2 types of sodium channels, a fast and a slow, and I also have a calcium sodium channel. The net effect of that is that the action potential is actually slower. And the reason for this is so that I only propagate that electrical signal in one direction, and I have sort of a long refractory period so that I can't propagate the signal and try to contract and flow blood in the opposite direction of what I want it to do when I'm squeezing the muscle and making the blood flow out of my heart. And we'll talk in detail about this when we talk about cardiac physiology. So let me give you an example. Let me ask you a question. You may have heard of botulism poisoning. Clostridium botulinum is a bacteria. It reduces a toxin that inhibits the release of acetylcholine. So which of the following symptoms would not be expected if you ingested botulism toxin? So hopefully you saw that in order for you to have some effect of botulism on this, you have to look at what of the muscles groups actually are involving acetylcholine. There's only one, and those are the skeletal muscles. So the skeletal muscles are necessary for you to breathe, for you to move your eyelids up and down, for you to walk, but they don't have anything to do with dilation or constriction of your pupil or moving things in a retrograde fashion through your gastrointestinal tract, i.e. vomiting things from your stomach. These are under the control of smooth muscle, which does not use acetylcholine, so they would not be affected by this botulinum toxin. So that's all for this small segment. I'll see you in a little while and we'll talk about pulmonary and cardiac physiology.